gonna be gone Friday night, Saturday night, Sunday. I'll be back on Monday. Okay? So, I'm gonna be in a place where I don't have communications. Okay? So if you have an issue, I'm not ignoring you. I just can't, I don't have access to my computer and I'm in the field. So, yeah. So, it's not, when I get back on Monday, I'll respond to everything. But I think you're pretty well set up to do stuff. You start on your next set of cards, right? Okay, I'm going to introduce one more thing since we do have a long weekend. How many are meeting with study groups? Okay, raise and proud. Okay, all right, good. That's not good for you. Is it helping? Yeah, okay, it's helping. Yeah, find a study group. Find a study group. I mean, I'll sign this as homework if you don't do it on your own. Okay, find a study group. You've worked with people in the lab long enough. You've got people that are, you sit by. You've got study buddies that we work with. Find a study group. No excuses. Okay. All right. Find a group. And you're like, oh, I'm so shy. Again. Okay. You got to do it. Why? Because when you're in your head and you're all by yourself, in a medical team, we never send out one person. We never send out one person. Why? We want 3 sets of eyes on a patient. We want 3 sets of eyes. And even in TCCC, when we do tactical combat life-saving, okay, we go in pairs. We go in pairs. Why? Because my partner is going to see something that I don't see. Okay? What makes studying any different? It doesn't. Okay? So I would never send a medical team of 1 out, ever. I would get demoted. Okay? Why? Because you have to work as a team. You're a medical team when you're working. You're a medical team outside. That is your team. If you've got questions, I call my team. I've got them on speed dial. I got something I don't know how, what is this? I call them. There's no shame in that. That shows maturity. That shows wisdom. When you keep all your questions into yourself and you're like, well, I don't want anybody to think you're stupid. Guess what? We all are, there's all things we don't know. Okay? Even at this level, there's things we don't know. So one of the things you want to do is get with the team. Because everybody studies differently, right? Okay? Everybody sees things a little bit differently. Their mind maps will be different, right? Okay? Bless you. So, so you want to make sure that you get a different set of eyes. It helps with the connections up here. Okay? So when we come back here on Wednesday, 1 week from today, I want to see everybody's hand in the air. Otherwise, I'm going to start assigning you. That's how important this is. Okay? All right? Please, please, please get a study group. And a study group is 2 or 3, right? 2 or 3. You've got the same people you're working with on cards, right? Okay, some people have dropped out. Some people have, come in. Figure it out. You're in a social environment, so figure it out how to do that. Okay? It will help you. It will help you. And you're saying, oh, I don't have time. That's a red flag to me. If you're saying, oh, I don't even have 1 hour that I can meet with somebody else to help get my skills up, that tells me you are overcommitted. Okay? And those that said, oh, I can do 50A and this at the same time, or 50B and this at the same time, or I can take 12 hours and have my 10 kids and everything's going to be fine and I can do everything, we're starting to find out that's really not true, is it? You can do everything, but not all at the same time, right? So yeah, so on Wednesday, I want to see everybody's hands in the air that they had met with the study group, okay? Is that cool? You got a week, okay? How many are going to go to SI today? Okay, go to SI. She talks about the practical, so yeah. All right, good. Okay, so let's do this. We're going to do a chalk talk today, so unless you're taking notes on your pad, I don't want to see computer up, okay? I don't want to see computer up. So some of you are taking notes on your iPad, that's good. Shut it down. Yeah, off, you don't need it. Okay, well you should have some paper or something to write with and somebody you can sit and talk to because we're going to be doing some things here. So if you're not sitting next to somebody to work on stuff, take a moment and move so that you can be by somebody so you can discuss. Okay, you ready to go? Okay, colors, get out all your good stuff. We're gonna get serious. New piece of paper, brand new piece of paper. Okay, you and your study buddies are gonna be working together. And I want you to build a mind map with this, DNA. Okay? Okay, no notes from memory. Everything you remember about DNA. This is from the top of your head. What do you remember about DNA? The more complex, the better. All the DNA details, yes.
All on our own paper, right?
All on your own paper. Yep. All on your own paper.
That's made out of double helix structure.
Yeah, for eukaryotes, right? Isn't eukaryotes is the
I think it's all of them. Because I think single strand is RNA.
Okay, you should be writing your own mind maps. You can talk about it with your group in just a minute. But for your own mind, what do you remember about DNA? Okay, get with your study buddy and see what you're missing, what they came up with, and make sure the information is correct. If you're not sure, ask. Are you sure this is correct? Okay?
I guess this is the circular, and there's plasmids. They can like exchange info between other bacteria. Eukaryotes, it's linear, and it can condense protein.
Yeah, no, I mean, that's pretty much it. The only other thing, I'm like, I don't like things, but like, I forgot to write histones for DNA too.
That's only in eukaryotes.
In the eukaryotes.
But you guys don't remember when you were like parties and I was like celebrating my birthday? It wasn't even my birthday. I think yours was on the 19th. Huh? Oh, it's also replicated during S. You need to do it. Yeah. Yeah, sometimes.
Honestly, I didn't have much to write. The only other thing I wrote was like biotech, but even then, it's like I didn't really have anything to write about it.
Okay. All right, let's see what you got here. Now, if I were doing this, and this is my crazy brain, I'd be thinking about history. I would think about the nucleotides. Okay. All right, and I'd be thinking about replication. Okay, how do we do this? And then, yeah, so I'd start there. Okay, and what are the nucleotides? Thymine. What? Okay, thymine. Okay, so. Let's add some to the nucleotides. What are all nucleotides made of? What are the three components?
Nitrogen space.
Okay, you got a sugar like this. What kind of sugar is this?
Deoxyribose.
Deoxyribose. It's got a phosphate group and the base, or the nucleotide base is it? Okay, so those are the three things. Okay, and then these get switched out. Now, if I asked about other nucleotides, there are two other nucleotides that aren't part of DNA. What are they? Uracil and ATP. Right, uracil and ATP. Okay, you might want to do a different mind map. You want to add to your mind map, but get your pictures in there, just like the video showed. Okay, get some pictures in there. Okay, then when you look at all of this, we know what is known as Chargas rules. What? C binds with G and A binds with T, right? And hopefully you've got GCAT, puppy. Have you seen this before? Okay, GCAT, puppy. This tells you the purines is G and A, and the pyrimidines are C and T. So GCAT puppy, you don't have to remember, you just have to remember GCAT puppy. You can always tell which ones are pyrimidines and which ones are purines. Okay? All right? Some people had, okay, it's a double helix. Okay, let's talk about the bonding that's here. What is the bonding that's in between here? It's hydrogen bonding. Good. What is bonding along the backbone? What kind of bonds are those? Phosphodiester bonds, which are covalent, right? Okay, that's good. Does anybody have anything else in terms of the individual nucleotides that is missing from this section? You've got anything additionally? Okay. All right. Okay. Let's go with replication. I'm going to take this down. Okay. Let's go with replication. Okay. And this is, you had it in Bio 1 and 50A, so, Okay? Tell me everything that you have about replication that's on your thing. Okay, really loud. It uses helicase. Okay, so we've got 3 enzymes that we use. One is helicase. Okay, what are the others? What? Okay, DNA polymerase, DNA pol 3 for bacteria. Okay, what else? Hold on, what are the 2 enzymes? What? Ligase. Okay, we're missing one. What is it? Come on, somebody's got to know this. You read the chapter. I hope you did. Okay. What's the one here? Well, there's actually 7 polymerases for DNA polymerase. So, yeah. So there's polymerase DNA pol 1, 2, 3. They all have different functions. Okay, I'm going to leave this blank for right now. Okay. All right. What else do we know about DNA just from Bio 1 and 50A? What? Okay, so when you replicate, you have a leading strand that replicates continuously, and you have a lagging strand, right? Okay. All right, keep going. What else do you know? Yeah. Go ahead.
Is 2 primates?
Yes. Good. Good. That's good. Primates. All right. Yeah, it's primates. Okay. All right. What else about replication? Okay. So if you look at this, it's going to happen in S phase. Good. What else? Ooh. What is 5' to 3'? Okay, what happens 5' to 3'? Okay, so I'm hearing 2 things. 5' to 3', okay, this is the direction things are made. It's read 3 to 5. Okay, it's read 3 to 5 and made 5 to 3. And then some people are saying, oh, they're different directions. So if you've got 3' here, well, you should have 5' here, right? This is all Bio 1 stuff, right? All anatomy and Bio 1, okay? All right, what kind of replication does DNA have? There's a name for it. Who said it? Okay, one more time. Good. Semi-conservative. Okay, semi-conservative replication. Okay, this is a good time to look at your overall mind map. Okay, some of you got really focused, and that's excellent. Some of you went off the grid. They started talking about RNA and this and translation. Is that that's way off. Way off from what we're doing. If we were talking about the central dogma, that would be important. But if we're just talking about the DNA molecule, that's it. And I understand a lot of people are trying to jog their memory and stuff, but it also could be a sign that maybe you don't remember some fundamental basics too. And that's what we're trying to get in this lecture, is the basics of DNA. Okay, and we're building on things that you've already learned. Okay, all right. And do you have anything on here that is there anything missing from up here that you have on your sheet? What? What kind of structure are we talking about? Oh, okay. So let me do this. Let me draw it up here because this is important. Okay, so O, like this? Like of the nucleotide?
Yeah.
Okay.
Like the O and then OH
care about this and this. Okay, and then you've got the carbon, the thing here. Okay, what carbons are these OHs at? Remember how to number carbons. Okay, this is carbon 1, 2, 3, 4, 5, right? Okay, we're going to draw a lot of the 3, 5 to 5 prime in a little bit. Is that where you're getting at? I'm trying to read what you're... Yeah, okay, we're going to get there. Yeah, we'll get there. Cool. Okay, now, how many of you have electronic books? How many have electronic books? Okay, a lot of you have electronic books. Okay, how many have the paper books with you? How many don't have your book at all? Okay, okay. What I'd like you to do with your partner is get with your book, whether it be paper or electronic, and find this section, okay? Nothing else, you're not messaging people, whatever. We're looking for the section where this is talked about, okay?
Just need to find where they're talking about it. Which chapter 9?
Okay, read through the section with your, whoever's working with you, okay? On some of the DNA things, just on anything we went over as far as the structure of DNA, okay? And if you're really going to be an efficient student, in your mind map, I put the pages, so you don't have to keep searching for it. It's taking, you know, I'm looking at you, it's taking over a minute to try and find stuff. And, you know, when you're studying and you're cramped for time, you don't have minutes to waste because you're disorganized. So put on your mind map, put where the pages are so you can find it very quickly. Okay, so you've got your mind map in front of you. Where can you find this stuff? So when you're researching, or when you're looking for things, you can find it really quickly.
That's like chapter 9, right?
So your whole goal right now is to find the things that are missing from our mind map that have to do with DNA structure. Where's your throat? Oh my gosh. Yeah. That's why I hate electronic books. Because boy, if the power goes out, you're stuck. You have no Wi-Fi, you're stuck.
It starts at page 293.
Okay.
Chloroplast and mitochondria have their own.
Or if you want to go by the index, then it's page 260. That's where it starts.
Okay, what about the structure of DNA? Has everybody got there? Is there any, read through that section, and, you know, did we miss anything? Okay.
section's part of this section.
What did we miss? Is there anything missing as far as the structure? Yeah. Structure. Not yet, because that's under replication. You're jumping the gun, so hang on. Yeah. Okay, we didn't talk about chromatin fibers. Okay, that's good. So what's the difference? When we talk about chromatin in prokaryotes? Okay, what form does the chromatin take when it's what? It's a circle, right? But in eukaryotes, our chromosomes are what? Linear, and we have histones to help with that, right? To wrap around there, okay? So if you want to include that in your mind map about the difference between prokaryotes and eukaryotes, that'd be good. What else do prokaryotes have? They also have plasmids. Our cells don't usually have those plasmids. Okay, good. Anything else? Yeah? Yeah.
Purines are double rings, with some primed or recessive.
Okay. Pyrimidines.
Pyrimidines are single.
Okay, good. Let's add that. So when we did GCAP puppy, okay, purines are what?
Double ring.
Okay, they have 2 rings. The pyrimidines have 1, right? Okay. So you can also remember 2, 1, 2, 1. Yeah, okay, good. Excellent. Anything else? What? Oh yeah, that's going to be replication. So we'll talk about that. This was just about structure. Okay. Cool. All right. Is this how you're going through your notes when you study? Okay. This is a good exercise to show you how you should have all resources there that you're going through this and studying. Okay. Is that cool? All right. Okay. Set your stuff aside. Let's take a look at some of the rest of the things we're going to look at today. Okay. Don't move from your groups. You're going to be pull that desk around. Yeah. Okay. Pull that desk around. Okay. So now I'm going to ask you, how many read all this stuff before you came in? How many did? Okay, if you had read this before you came in, when we started doing mind maps, would it have come quicker? Yes. Okay, so now you've got some data to show that if you are priming your mind by reading before you come in, okay, then, that's it. And it helps you, it's your second time through. Okay? All right, please set aside all your electronics, everything. I want you to take paper notes as we go forward, okay? So, so set aside everything. You should have a piece of paper. You're going to be drawing, okay? So off we go. Okay, so who are these two suspects up here? Who are they? Okay, one is Watson and one is Crick. They look the same to me. I don't know. I really don't have much, yeah, we have to study them because they're part of the history. Okay, definition, general definition we're going to start with. Remember this, genotype versus phenotype. A genotype gives you the sequence of DNA, right? That's your genes. What is in your genes? Your phenotype is what you see, what is actually expressed. Okay, so let's draw out the central dogma of DNA. Okay, DNA goes to mRNA, goes to protein or polypeptides. It says a circle around it because DNA is self-replicating. Okay, this little arrow is for, transcription. Good, transcription. And this arrow is for translation. Okay. Which of these things happen in the nucleus?
Transcription.
There's 2.
Oh, replication.
DNA replication and transcription. That'll happen in the nucleus in eukaryotes. In the prokaryotes, it just happens out in the cytoplasm. That's okay. All right, we're going to keep coming back to this as part of the central dogma, and this, for a lot of you, could be the basis for another mind map. Okay. So, let's look at a couple of things here. Okay, let's look at a couple of genomes, and you might want to have your mind map handy so you can make notes in terms of the size of the genome. Maybe you don't have a bubble for the genomes. Maybe you should. Okay, viruses, these are small genomes. And when I say genome, by definition, the genome is the entire genetic material that is in that cell. That is a genome. Okay? The entire genetic, the whole complement of genetic information that's in that cell. Okay? All right. That's what a genome is. Okay? And for some of these viruses, it's 4 or 5 genes. But, our genes, we've got over 20,000 genes. Okay? Bacteria, we've got cytoplasm and plasmids. That encompasses the genome because you can't just have the bacterial DNA, you also have to include the plasmids. Okay, so when you talk about plasmids in bacteria, you have both. Okay, and if you're looking back at your exam 1 and you're like, oh, I missed that little definition, I'm screaming about it, literally, over this because these are the little definitions that makes a difference between a B and a C, right? That's really the definition. So, okay, eukarya. All of these are linear chromosomes. What do the linear chromosomes have that the circular chromosomes do not? Histones, right. And you get circular chromosome in mitochondria. Okay? It has 37 genes on it. Okay? Most of those genes code for things like the electron transport chain, okay? The things that have to do with cellular respiration. I'm going to pass out if I keep going at this pace. Okay. Not good. All right, let's try this again. I put in a work order, but really did anything. Okay. All right. Any questions on this? There's one other cell organelle that has its own DNA. Which one is that? Chloroplast, right? Chloroplast, excellent. Okay, good. Okay, so we've talked about the structure of DNA, double helix, and all that good stuff. So let's draw this out so you can actually see it. Okay? When we talk about DNA strands being anti-parallel, okay, where we've got 3 over here, 5 over here, it's referring to I like to look at it as which way is a house facing, okay? So if we're going to do that, okay, so we have a little house here, OH, here's the nucleotide there, here's a phosphate, and it's going to bind to the sugar on top like that, and here's another thing, and here's the phosphate, okay? All right, so if you remember your carbons, this is 3 and this is 5, right? So this strand is going in the 3 to 5 direction. All the houses are upright, right? All the houses are upright. Does that make sense? Okay, is this the question you were getting back to, is where does all this stuff go? Yeah, okay. Yeah, okay, so if these strands are anti-parallel, what has to happen? On this side, which way are the houses facing? Down. Yeah. Okay, you still got the phosphodiester bonds. Oh, wow. Yeah, let's get it a little further apart. Okay, there we go. There's the phosphate. And then, yep. And the hydroxide is here. So the 3' hydroxide is on this side, and the 5', which is attached to this carbon, is there, and I'm missing a flag. Okay? And you've got hydrogen bonds between these, right? Okay, let's make this an A and a T. G, so it needs one more. Okay. All right. So when we look here, it's opposite. Does that make sense? Okay. So if it's 3 to 5, all the houses are upright. If it's 5 to 3, all the houses are upside down. And what is the term to describe this? It's what? Anti-parallel, right? Anti-parallel. Okay. So far okay? Right. It's easy to look at the diagram, but when you draw it out, it's a lot harder, isn't it? Yeah. So I'll wait a little bit and get caught up on that. Is that good? No, it's not good because what? Oh, who said it's not good? You're not done. Okay, good.
No,
Yeah.
good, but not done.
We're good, but not done. Okay, so that's okay. Good. Tell me if you're done. You know when you're done. Because I can look at these pictures all day. They don't make any sense. But once you draw it, you see whoosh, how they're anti-parallel, right? Okay, good. Good so far. Okay. You all done now? Okay, good. All right, that's cool. All right, here's the structures. Are you going to have to draw the bases? No, but we've already talked about here. Now, if you were going to use ChemSpeak, how you should be able to describe the difference between deoxyribose, which is in DNA, and ribose, which is in RNA. Okay, and when I talk about ChemSpeak, what carbon do you think are things different here? The carbon 2, right? So here's carbon 1, here's carbon 2. Deoxyribose, it's missing an oxygen. In ribose, it's got an oxygen. It's got an OH group. Okay. All right. And we did GCAP puppy, so that's good. Okay. All right. Oh, we did this too. Purines and pyrimidines. Okay. All right. See if you can, from your memory, don't look at anything, draw a nucleotide. Just from your memory. See if you can do it. The basic structure of a nucleotide. Don't look, just do it from memory. Okay? Do the best you can from memory. And then see. If you go and look it up, you're not building the memory thingies. What are the 3 things that a nucleotide should have? Nice. Okay, what are the 3 things that a nucleotide should have? Sugar. Okay, so you got a sugar, okay, the satellite dish, the phosphate, and then the flag, which is what?
Nitrogenous base.
The base, yep. And if it's DNA, what's going to be on the 3rd carbon? OH, okay. There's a carbon here too, but we just draw the line because it's the phosphates there. How many of you got this from memory? Good, good, excellent, all right, good. Let's go on. All right, we talked about this, we talked about anti-parallel and complementary, we've talked about we've talked about that. Okay, so this is really kind of cool because there's so much redundancy in this molecule. It's very mistake-proof, as best as can be, but you do get mutations. And what's the definition of a mutation? What's a mutation?
Change in
a permanent change in the DNA sequence. It's not just a change. You get changes all the time. You go out in the sun, you got a ton of changes. Okay, but most of the changes can be repaired. That's not a mutation. A mutation is a permanent change. Okay, is that cool? All right, so, all right, now we're gonna shift gears here. Okay, we're gonna shift gears and talk about experiments. And I don't know, did anybody on their mind map put experiments that gave us DNA? Okay, so you might want to do a new section on this, okay? And look at the experiments. All of these experiments we talk about, they won Nobel Prizes, okay? Do you know about the Nobel Prize system? Okay, every October, the first week in October, it's so exciting. I can't wait because they're going to announce who gets the Nobel Prize. And they have different prizes, right? They have economics, they have physics, they have medicine and physiology. They don't have a biology one, but they do have chemistry. And then they have literature, and then they have peace, right? Okay? And what do you win? You get a pretty cool medal and about $1 million tax-free. I'd want to win that. That's pretty good. I'd take that. Okay? And then, what else? Yeah, so they're named after Alfred Nobel, and he, does anybody know how he got his fortune? From TNT, from dynamite. Okay, and they don't give one in mathematics. Do you know why? Because his wife ran out for the mathematician, so they don't give one in mathematics. Yeah. So instead in mathematics you have the Fields Medal, which is given out every 4 years. But yeah, so these Nobel Prizes are pretty cool. My teacher that taught me genetics, it was a Nobel Prize winner. Okay? His name was Mario Capecchi, and he did a lot for the Foundation of Genetic Engineering. You can look him up. He was a terrible teacher. Okay, I gotta tell you. So when I was taking genetics and biochemistry, which he taught both, the lecture hall was like 400 people in it. And he walked in one day and said, well, I couldn't decide whether or not to go to the basketball game last night or to prep a lecture. The Jazz won. And then he walked out. And he's like, exam next time. And it was we were doing electron transport training in Krebs. Can you imagine if your professor never said a word about it? Okay, there's an upper division biochem class. That's the one he pulled this stuff. But if you got him in his lab, that's where he loved to be. He hated to teach. He loved to be in the lab, right? That's where the magic was for him. Okay, great guy. I wrote him when he got the Nobel Prize, and he wrote back. He didn't remember me because I was one of those people who sat in the back and got a C. Okay, if that. And he said, I told him what, we were talking about him in class, and he said something like, light the minds of the youth on fire. And I'm like, that's a felony. So anyway, he was pretty cool. Not really. I mean, I never want to have him as an instructor, but in lab, he was fantastic. Okay, so we're going to talk about how in the world did we determine that DNA was a molecule that actually passes on traits so you can actually see the phenotype. Okay, now when was DNA discovered? I mean, when was it published what the structure of DNA was? Any idea?
50s?
Last month? No. When was it published? It was published in the 1950s. Okay, it was published in the 1950s in Nature. And yeah, so a lot of these experiments that we see happen way before that's published, right? We're talking 1928. Huh. All right, let's see what they did. Now, in order to understand this experiment, the key is, as you're taking notes on this, is the difference between a smooth colony and a rough colony. Do you see the difference? Okay, what's the difference? Okay, the rough colony is rough. The smooth, the boundaries are rough, But look at that smooth, okay? Why is it smooth? Because these have capsules on them, okay? So it makes the colony nice and smooth, okay? So which is harder for the immune system to attack? Just based on what you see here, which is harder to attack? The smooth one, because it's got capsules on it, makes it nice and smooth. This rough one, easy, okay? Doesn't have the defense. So let's see how it is. Let's inject this little mousie with R strain, which is R for rough. Does the immune system kill it off? Yeah, it lives. If you give it S, what happens to the mouse? Yeah. What did you say? Unalives, okay, all right. All right, so yeah, it's gone. And when you culture, you find S strain in it on autopsy, okay? Now, so which one is your control here? Trick question, they're both, okay? They're both your control. You gotta see what the S strain does, you gotta see what the R strain does. You got that in your head. It's good? Okay. Now we're gonna take the S strain and we're gonna heat kill. What does heat do to proteins and enzymes and stuff like that? It denatures. It also denatures membranes and things like that. So is this gonna be a viable, livable S strain? No, you inject it into the mouse and it's healthy, okay? Because why? You denatured a lot of it, okay? Now, this is a funky one. Good. So, here's the living R strain and dead S strain bacteria. It's been inactivated. It's not viable. But you put it in a flask, mix it up, inject it into the mouse, and it dies. Would you expect that? No way. Because what's the only living cell here? R. So something had to happen in that flask. And when I talk about a flask, I'm talking about, you've got like a big flask here and it's in a broth and you've got R and you've got S in there that's been heat killed. Now, if it's heat killed, okay, what do we know? Is that membrane going to be perfectly intact? No. Okay? So what in the heck happened here? And if you read before you came in, you know the answer like that. What happened here? There's one word that explains everything that happened here. What is it? No, it's not mutation. What? Transformation. Good. Did you really? Huh? That's okay. You read the summary, right? Yeah. Okay. Yeah, transformation. What in the heck is that? Well, guess what? If you've got S bacteria, okay, and it's got holes in the membrane here, okay, and we got the rough bacteria here, okay, is it possible for DNA to go in here and enter in through like porins or something like that that's in the rough? Yeah, and that's exactly what happened. Is the DNA that was in S goes into R, and what did it do? It transformed the phenotype. And we know that, how? Because on autopsy, what do we see? We see S, okay? All right, in 1928, this must have blown their minds. But do you see how transformation, what is the thing that has to happen for transformation to occur? What's the key? You've got to have DNA from the S make it into the R so it can be expressed, okay? That's the key. Okay, is that good? Okay, 1928. If I got these results, I think I might have gone to the bar rather than, think I screwed up. But no, he got a Nobel Prize for this. Okay. All right. Another experiment here. Avery, McLeod, and McCarty, 1940s, almost 20 years later. Okay? And now what they do is they're going to use some enzymes, protease and DNase. How do we know those are enzymes? ASE. Okay, so they do the same thing. They get it in here. except they've got 2 flasks going, and in one flask they add protease, and the other one they add DNase. If they add, okay, so the same thing, but if they add the protease, what's missing from the cell now? What do proteases dissolve? What does the name tell you? Proteins. Yeah. So now you've got proteins that are getting dissolved, and guess what? The mouse dies. But when you give DNase, what is DNase going to dissolve? DNA. Oh, what did you stop? You stopped transformation. Yep, you stopped transformation, and this mouse lived. Did you stop transformation here? No, because proteins don't really have much to do with transformation. What's the key? DNA. Okay, it's the DNA. Okay, then along comes Edward Chargaff, which is Chargaff's rules. A binds with T, C binds with G. Okay, and then this is the critical experiment that shows that DNA is a genetic material. And it happened in 1950. And why is 1950 so important? What's happening in the world in 1950? What? We just got out of World War II. How did we end World War II? With a nuclear weapon, right? Sounds like, no, it doesn't sound like this at all. Yeah, with a nuclear weapon. And when we were developing all that nuclear technology, one of the things that we developed was a way to use isotopes as biological tracers. This changed medicine. I mean, the bomb's a horrible thing. It's a horrible thing. But we use radioactive tracers now all the time in medicine. If you've had a CT with contrast, if you've had a barium test, if you've done PET-CT, you've all had, isotopes. So there's a couple isotopes you have to be aware of. One is P32, that's how they pronounce it. They put the P first and then the 32. 32 gives atomic weight, and they give S35, and that's sulfur. That's one of the isotopes of sulfur. Okay, for P32, we use this to trace DNA and RNA because of that phosphodiester bond that's in the backbone of DNA and RNA. For sulfur, we use this to trace proteins. Okay, you good so far? All right. So, what's going to happen? Tell me what you remember about T-even phages. Off the top of your head, what do you remember? We talked about these last time. T-even phages, what are they about? They're bacteriophages. They look kind of like, some kind of space monster. Okay, like this. They have tails here. Okay, so tell me what this part is called. The capsid, and what is it made of? Proteins, good. And what is in here? DNA or RNA. Okay, it's usually DNA for this. Okay, so let's use our isotope. Everything that you see is red is labeled with P32. Everything you see is blue is labeled with S35. Okay? Now, what this experiment was trying to show is what actually gets into the cell to change the phenotype. We can show it indirectly with proteases and DNases, but we needed the tools of isotopes in order to do this. So let's see what happens here. Okay, so we've got, here's a E. coli. Okay, E. coli. All right, and let's pretend this phage is going to land on the surface of E. coli. We know that there's hundreds that do, but this one in particular. Okay, Now, in order to change the phenotype, the molecule has to enter the cell of E. coli. What's the only thing that's going to enter the cell here? What's the only thing? We talked about this last time. What? What's going to enter the cell? DNA, right. We know that. We know DNA is. So what isotope should we find inside of E. coli? Yeah, P32. And guess what? That's what they found. All the S35 was outside the E. coli, so we know protein doesn't influence, transformation. What is it? It's the genetic information. Okay, does that make sense? Okay. All right, what are your questions about this? This is such a cool experiment. And I think drawing it out is a lot easier than trying to interpret this. This shows the same thing, actually. But, yeah. I think, does drawing out help? You see the process? Okay. So, Hershey and Chase. Hershey and Martha Chase. Martha Chase, yeah. Okay. Any questions on Hershey and Chase? Again, Nobel Prize, big medal, $1 million. Okay. I'm not sure. I'm not sure if they shared the prize. They might have. I don't know. Have to see. Okay. So, all right. So there are 4 scientists up here that deal with the structure of DNA: Franklin, Watson, and Crick, and Maurice Wilkins. Okay. Let's draw these actors out so we can see what they're about. Okay. All right. So here's Watson, and here's Crick, and here's Wilkins, and here's Franklin. All right. Now, the reason I'm drawing all these out is because of how they came about everything. So he was the PI, the principal investigator. She was a postdoc. This is what you do after you get your PhD in science usually, is you go and do a postdoc for several years. Some people do a postdoc for 20 years, okay? Sometimes you don't do a postdoc, you get a job right away, okay? It just depends. But she was a postdoc, okay? These were both staff scientists, so they were doing science. So this is scientist, okay? All right, and they were all trying to figure out what the structure of DNA looks like. Watson and Crick had the double helix down, but they had the bases going on the outside. Bless you. Okay? We know that's not right. They knew it wasn't right. The math wasn't working. Okay? So they tootled on over to the office of Dr. Wilkins and said, hey, we know that your postdoc, Rosalind Franklin, she's doing some X-ray crystallography. What is X-ray crystallography? She would crystallize the DNA and then run X-rays through it. And when you X-ray something that's solid, you get a pattern. When you X-ray a bone, you get a pattern, right? But when you give X-ray a 3-dimensional molecule, you get a really unique pattern that you can actually calculate the angles of it. So that's what she would do. She would be in her lab all day and be using X-rays, and eventually it killed her. Okay, killed her. So Wilkins, okay, said, I've got this interesting photo. It's called Photo 51, and I can show it to you. Now, that's bad practice. When you collaborate with people, you let everybody know that everything's being shared, right? That's what you do. Otherwise, you can end up in court or something like that. I mean, like the collaborations that I do, everybody knows everybody else. Everybody can write their contributions of what they've done, but there's no sneakiness going on. And that was really sneaky to let them see her data without her permission. That was her data, okay? So as soon as they saw that picture of Photo 51, they went, crap, we've got it wrong. And they said, the bases are on the inside. Well, guess what? She had already figured this out. The bases are on the inside. Okay? So they made a pact that 4 of them would publish the 2 papers. It's only a page and a half in Nature. Watkins and Crick came first, and Wilkins and Franklin was a page behind. But then it came time for the Nobel Prize. They don't give Nobel Prizes to people who die. She got cancer, probably from all the x-rays that she was using, and she died at the age of 34, I think it was 34. Okay, 34. So she died, she never got her Nobel Prize. The other 3 did. Okay, isn't that sad? I think it's one of the saddest stories in science. Okay, but yeah, so that was her, and that's her photo 51. Okay, they didn't do the experiment, she died doing the experiments. Okay, and that sort of pisses me off. Okay, what? Go ahead, what?
Why did she have to be?
Yeah, why did she have to be the one? You know what, she carried, I don't know. You know, there's a lot of those questions floating around in the universe. And then, you know, lately Watson and Crick have gone off their rockers and they've said a bunch of stuff that is crazy. So, I mean, she was one classy lady, so, yeah. Okay, all right, so, right now, we've had a number of experiments. Write them down. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Write them from memory. Okay? Write them from memory. What? Just the names, maybe one word that says what they did. Okay, check with your partner. Okay? I think I counted 7 experiments. 7.
Yeah, I only got the 2.
Check with your partner.
The DNA gets into the bacteria and not the capsid that was last talked about.
I have the Griffith one where he found out that there was some kind of genetic barrier through the RNA exchange, and then Hershey found out that DNA was the carrier. And then Rosalind finding the specific double helix structure. That's not like
Okay, did you all get Griffith? Y'all got Griffith on your list? Did you all get Avery, McLeod, and McCarty? No. Okay, Chargaff, Hershey and Chase, Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick. Okay, you got those? All those? Yeah.
So I got a question for Watson and Crick. How did they know it was a double helix if they did no experiments?
They were taking other people's data.
But in general, not just from Franklin? Oh, okay.
Yeah.
Gotcha.
They're like, oh, what are you doing? Oh, look at there, do some math. Oh, yeah, that's exactly right. Yeah. Are there people that do that in science today? Hell yeah. Okay. Yeah. Yeah, we do that. I mean, you look at, like, I was thinking about this the other day because I got a call from an attorney, but yeah, so if you have a patent on something, usually the attorneys, you need a patent attorney to do it, and then part of their income comes from going after people that are infringing on your patent. And so, I don't see any of that, but they'll see it, and I'm like, oh, yeah, sure, if they're stealing our ideas and making money off of it, yeah, go after them. Yeah. Okay, we can't really do this here. I mean, now we do. I mean, some labs don't share anything. Sometimes you go into a lab, like when I was at Oak Ridge National Lab, I had to sign a non-disclosure agreement that I could never talk about anything that I work on. Okay, and when I did some work for SpaceX, same thing. Yeah, SpaceX. I helped to build a rocket in 2019, right before COVID. It launched in 2020. Yeah. I don't sleep. I teach and I think about all these crazy things, and then I'm like, oh, this would work. So yeah. On rockets, they have when you want to get to a certain altitude, they have what's called an air brake on it. And so me and 3 students designed this air brake. So yeah, they've got it. So we can't really talk about it, but air brakes on rockets are well known, so it's not a big deal. But yeah, that's what they do. It's not like I'm sharing plans to invade a country. I know, my phone was going off at 5 this morning. It's like, yeah, Colonel, we saw this. They released the train. I'm like, oh, shit, man. Okay, so, all right, let's talk about DNA replication. Okay, DNA replication. All right, binary fission. Okay, this is what the the prokaryotes do. With binary fission, this is really kind of cool. So you've got a circular chromosome, and this is key. All replication starts at the origin, and oftentimes they abbreviate it as ORI. Okay, just so you know. So if you're reading something online and you see ORI, that stands for the origin. They get lazy and they just put ORI, okay? This starts at the origin. Prokaryotes have one origin. How many do we have when we go through S phase? We have thousands. We copy, blow your mind, okay? We copy this much DNA in 1 hour, okay? Per cell. This is in 1 cell, okay? We have 1 hour to do it in S phase because everything else in the cell has to really shut down in order to do this, okay? So we're gonna have thousands of origins of replication, okay? Because our genome is huge, all right? Is that cool? Okay, so we know this, DNA is semi-conservative, so if we want to replicate a molecule like this, by the time we get done with replication, we have 1 old strand and 1 new strand. There's always a template strand, which are the black strands, and the non-template strands, which are red. Okay, is that good? Okay, replication forks, blah, blah, blah. All right, boring, let's draw. Okay. All right, you've got these, you should have on your paper these 4 enzymes. Helicase, primase, DNA polymerase, and ligase, okay? Now, I'm gonna play with this string a little bit, and I'm gonna wrap it up like it was a DNA helix, so it's gonna wrap up here. Takes a little bit. Are you strong? No? Are you a little strong? Okay. The partner says yes, it is. Okay, I've got a double helix here. It's really thin. I want you to pull it apart. Pull the strands apart in the middle. Okay. Oh yeah, see, you're strong. Look at that. Look at that. Okay, stop. My okay, what happened up here and what happened down here? Okay, see how, oh, you did a good job, thank you. See how tight this all is? This is a string. It works for a string. Will it work for a molecule? No. Okay, the molecules are too fragile. So when you unwind like that, you better have something here and here relieving the pressure to help unwind. What enzyme does that? Helicase. Okay, helicase. Now, there's different names for helicases. There's 1,000 different helicases. Some of them are called topoisomerases. Some of them are called gyrases. In bacteria, they're often called gyrases. Okay, there's topoisomerase 1, topoisomerase 2, topoisomerase 3. Why do you care about that? Well, if you're in oncology, some of the drugs target topoisomerase 1 and topoisomerase 2. You'll see that later on. Okay, but what do they all do? They fit under the umbrella of helicase. So what does it do? Okay, so if we were to draw this out, okay, here's our little DNA strand. The helicase will act as a swivel here and here. It requires ATP. And it unwinds and relieves the pressure on the molecule. Okay, so I'm going to pretend in my drawing here that I've opened up the replication fork here. And I'll make this 3' and 5', and 3' and 5'. Okay. And what's our rule? DNA is red 3 to 5, and it's made 5 to 3. Okay? All right. You with me so far? Here's our helicase. I'm only drawing like this part because we just need to see what's going on in that replication fork. Okay? Just imagine it's going on all over. All right, what enzyme comes in next? What's next on our list? Primase. Everybody forgot that except one person. So please remember primase. What does primase do?
It gives it like a start.
It gives it a start. It adds RNA primers, okay, to the DNA. Replication just can't start. It doesn't work like that, okay? So what happens is primase comes in and adds some here. This will be the leading strand, and this is lagging. Okay. Bing, 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 bing. Okay, real quick, what enzyme comes in next? DNA polymerase, DNA pol 3. It's going to copy, it will bind here, copy, bind here, copy, bind here, copy. Okay, and what are these fragments called? Good, Okazaki fragments. And then there's some enzymes, we're not going to talk about them, that come in and remove the primer. And you use the next enzyme that will link all this together. What is it? Ligase. Ligase is glue. It links those together. Okay, I think I have not put enough tutorials on this. I don't think I have. Okay, I'm going to add some more tutorials. So this could be, so you know how, oh, here we go. The bacteriophage, oh, no, I don't have them here. Okay, check out the tutorials. You should be able to, this is where we're going to start on Wednesday after the Quizzy Wizzy, okay? All right, have a great weekend. Email me if you need anything, just be patient. Out in the rain.
I don't know, man.